Okay, I'm gonna see if I can make that bigger. Let me move my head out of the way. All right, this is all about CRISPR. So CRISPR is a gene editing tool, which is awesome. If we can edit genes, we can fix problems in the genes. At least that's the hope or the goal. I don't know. There we go. Let's see if it'll blow up or not. Apparently not. Okay, so we'll go back to this one. It's actually bigger. Okay, so it's a gene editing tool that we have hijacked from bacteria, basically. So what bacteria would do is when bacteria would get infected with viruses, remember viruses are just basically DNA or RNA that's been given to something as an infection. So, and bacteria can get sick from viruses too. So when a virus would inject its DNA into bacteria, what the bacteria would try to do is try to find that DNA and cut it, basically rendering it useless. Okay, either cut it out and digest it, degrade it, or just cut it so that it doesn't work anymore. So if you cut a gene in the middle, then you're going to get the wrong protein. So that's how CRISPR is used in bacteria, was basically as a kind of rudimentary, basic, simple immune system. They don't have an immune system, so I'm using that term very loosely, but that's what they used it for, was to try to fight off viruses by killing their DNA. What we use it for is, well, let's back up for a minute. What they do is they produce an RNA that matches the viral DNA, and then they use that RNA and this enzyme called Cas9 to search for that viral DNA. So when they find it, and let's say that viral DNA is AAAAA, just to make it easy for ourselves. So their RNA is going to be UUUUU. They're going to search for that AAAAA. When they find it, this complex, this RNA enzyme complex, binds to it and cuts it. Okay, so that's how they do it. So what we've done is we've said, hey, that's a really good idea. It's very specific. It will find a specific gene because you build the RNA to match the gene. So what we are doing is we are saying, okay, you have cystic fibrosis. This is your mutation. If we can remove that, so you stop making the wrong protein and replace it with the right section of DNA, the right sequence, you will start making the right protein and you will no longer have cystic fibrosis. Okay. So what we do is we make an RNA that matches the mutated gene. Okay. We attach it to this CRISPR system, this Cas9 protein enzyme, and that just goes to work and it starts looking through the person's genome. And when it finds that wrong section of protein for that chloride channel, which is what it is, it's a membrane protein, okay, in the cell membrane. When it finds the wrong one, it attaches to it, cuts it out, degrades it, gets rid of it, and then we can then insert the right sequence of DNA. So, I mean, it sounds really simple. It's really complicated. We haven't gotten to the point of doing it with like people with cystic fibrosis and things yet, but that is the goal. It's being tested. It's being tested in animals first, making sure there's any wonky things that can go wrong before we kind of start doing human trials. At least last time I checked, there weren't human trials. There may be by now. You can Google and see. But that's the idea behind it, is to find or identify a specific DNA, cut it out, remove it, degrade it, and replace it with a healthy DNA. And in doing that, we could potentially cure all of these genetic diseases. I mean, think to your genetic disorders that you did and all the ones your classmates did. In theory, all of those could be cured. We are a long way away from that, but it'll be exciting to see where this goes in the next little bit of time. So that's just a basic of CRISPR. If you still have questions or just want to talk about it more or curious about it, let me know, and we'll go over it. Thanks, guys.